introduction and background of the DNA. DNA deoxyribonucleic acid is the hereditary material in human and all other organisms. Nearly every cell in living body has the same DNA. If you take the example of the man, you will find that 99% DNA is the same in all human body. Only 1% make variation. And if you see, the DNA is mostly found in the nucleus, and that is called your nucleus DNA. But very small fraction of DNA is also found outside of the nucleus, inside the mitochondria, and that is called your mitochondrial DNA. If you want to see the unique structure of the DNA, that is your double helical structure, and this was given by the Watson and Crick, and this double strand is made up of your sugar phosphate backbone, and that is attached with each other by a base pair, and that is made up of adenine, thymine, and guanine and cytosine, and all the hereditary information are stored in the code form inside this adenine and thymine. So who was the first person who isolated the DNA? He was the first Friedrich Missler in 1869. He was working in laboratory of well-known biochemist Felix Schuper and Seller, and he found that there's some chemical which are having a unique property, and he reported that was the DNA. Why it is important to isolate the DNA? It is the first thing important is to know the genetic cause of any disease, and you can develop the diagnostics and drugs for that. And it is also essential for the carrying out the forensic science and gene sequencing, detecting bacteria, virus from the environment, and finding paternity of any living organism.